MedCram.com.
Welcome to another MedCram COVID-19 update. The question that's on a lot of people's minds is, what kind of immunity do they have against SARS-CoV-2? So today we've got a unique opportunity to look into every type of immunity that you could possibly have against SARS-CoV-2, whether you were vaccinated with Pfizer, Moderna, Johnson & Johnson, AstraZeneca, Sputnik, Novavax, Sinopharm, even just gotten the SARS-CoV-2 infection. What kind of immunity could you possibly have against the most recent variant of SARS-CoV-2, which is predominant now across the globe and which is the BA.5 variant? And that's what we're going to look at today. Now, there's this paper that was published, and it's out of Dr. Wiesler's group at the University of Washington, and they've recently published this in the prestigious journal Science. And the article is titled Omicron Spike Function and Neutralizing Activity Elicited by a Comprehensive Panel of Vaccines. This paper actually goes into a lot of things, but one of the things that they go into specifically that interested me and probably you at this point is whether or not there is antibody activity against these different Omicron variants. And I think this paper does a really good job at answering some of these questions. You may notice in the author list some names like Alessandro Setti, Shane Crotty, who we've actually had on this program before. And of course, it's out of the University of Washington, which has a good track record here. And that's where the first case of SARS-CoV-2 in the United States first appeared. So if we go to the article, let's look at what they did here, because I think this is really interesting. They say here, we next assessed the plasma neutralizing activity elicited in humans by each of the 7 vaccines, and those are the 7 vaccines that we talked about at the beginning. But in addition to the 7 vaccines, they also looked at natural infection with SARS-CoV-2, and they evaluated the immune evasion associated with the constellation of S mutations present in the BA.1, BA.2, BA.2.121, and the BA.4/5 Omicron sublineages. Then what they did next here is they looked at how well the plasma from subjects who had each of these types of either vaccination or infection did against neutralizing these different sublineages. And the way they did that was they basically took the spike proteins from these sublineages and connected it with a vesicular stomatitis virus and placed it on a petri dish that had with it the Vero E6 and TMP-RSS2 cells to see whether or not the serum from these subjects could protect the cells from becoming infected with these specific Omicron sublineages. So basically what they were doing was an in vitro experiment to see how well antibodies from these subjects could protect against the various different sublineage Omicron and other variants of SARS-CoV-2. So when they look at the regular infection with SARS-CoV-2, specifically you should know that the plasma, which was obtained from individuals previously infected, were the ones in that area up in Washington where the first SARS-CoV-2 appeared in the United States, and that's known as the Washington 1-like SARS-CoV-2 strain. And as you may remember, this was occurring well before any vaccination of SARS-CoV-2 took place, almost a year before. So let's take a look at the data. Here's the data, and I've branched this out here below so you could see the data more clearly. At the bottom, we have exactly what type of serum we're talking about. And in this case, this is the SARS-CoV-2 infection, and we're looking at neutralization. And so the higher the number, the more immunity there is, the more antibodies there are, and the stronger the antibody response against the spike protein. So high numbers are good. And you can see here we have Moderna, we have Pfizer, we have Novavax, Johnson & Johnson, AstraZeneca, Sputnik 5, and Sinopharm. And specifically, each one are given in the doses that they are approved for. So Moderna is 2 doses, Pfizer is 2 doses, Novavax is 2 doses, Johnson & Johnson is 1 dose, AstraZeneca 2 doses, Sputnik 5 is 2 doses, and Sinopharm is also 2 doses. Now, unfortunately, they were not able to get the serum at each point along the infection. So specifically, what we're looking at here is the mean days after the event. And so for the infection, this is 41 days after the infection on average. So this is on average, if we look at the average subject in this category, how many days after the average infection did they submit their serum. For Moderna, it was 13 days after the second dose. Pfizer, 14 days after the second dose. Novavax, 82 days after the second dose. Johnson & Johnson was 79 days after the first dose, because remember there's only one dose with Johnson & Johnson, and about 30 days for AstraZeneca after the second dose. Sputnik 5 was around 60 to 90 days, all we have is a range there after the Sputnik dose, and then 69 days after Sinopharm. So what we see here in each category is the different types of variants. So the G614, as you can see here, was the very first strain that we had in the United States. And of course, we went through Alpha and we went through Delta and we came to Omicron. And so what we're looking at here is Omicron BA.1, Omicron BA.2, the 2.121, and finally the one that is current at the time of this video production, which is the BA.4 and 5. And the first thing that you can see here right off the bat is regardless of which category we're talking about, if you look very carefully across the board at each of these categories of BA.4/5, whether it's the infection or the vaccines, you will see that in general, there is less of a response against the BA.4/5 subvariant. Although there is still some activity. And so they've actually labeled this with numbers. And what they're looking at here is what fold reduction there is in the neutralization titers. You can see here from Moderna versus the G614, the original strain that was used in the production of the vaccine, there's about a 29-fold reduction in the neutralization ability of the antibody to specifically neutralize the virus. For Pfizer, with the Omicron BA.5, it's an 18-fold reduction. With Novavax, there is a 23-fold reduction. And some of these, we actually don't have numbers because of statistical significance. But you can clearly see that there is a numerical reduction in each one of these cases. I should point out that antibody neutralization is not the end-all, be-all of the activity of vaccination. For those following MedCram, you should understand this, and we've talked about this before. There are T-cell responses, and T-cells have a very robust ability to cross over and to manage these different spike proteins more than just immunoglobulins from B-cells. This is a surrogate for antibody neutralization. This does not necessarily mean that this contributes also into the ability of T-cells to do their job. And we'll talk about that more. And we've seen this over and over again with different variants. Even though the vaccines have been modeled after the original spike protein, there still is very good efficacy of the vaccines to prevent hospitalization and the worst outcomes of COVID-19, even though it may not be preventing infection. And that's why it's so important to understand the difference when we're reporting in the news media about the difference between cases and hospitalizations. Now, even there, as an aside, we'll talk about hospitalizations. You will see an increase in hospitalizations when you see an increase in cases. And the reason why is because every hospitalization in the hospitals that I work at are tested for SARS-CoV-2. So you might have people coming in with appendicitis or cholecystitis. These are basically completely unrelated to SARS-CoV-2 infection, and they might be positive because they may be carrying a subclinical or very mild form of SARS-CoV-2. So the question really is not if people are being hospitalized with SARS-CoV-2, but rather are people being hospitalized because of SARS-CoV-2? And I can tell you that whereas at the beginning of the pandemic, when we were seeing hospitals overflowing and putting 2 patients into an ICU room and having ICU nurses taking care of 4 patients rather than 2 patients, there was no question that people that were being admitted with SARS-CoV-2 were being admitted because of SARS-CoV-2. Now, that's as opposed to what I'm seeing today, at least locally in my hospital in Southern California, where we are seeing some people coming in with SARS-CoV-2, but they're not coming in because of SARS-CoV-2, at least not at this point as yet. Hopefully that stays that way. The point being that vaccines have much more to their abilities to protect than just neutralizing antibody. But nevertheless, I think it's an interesting exercise to look at this in vitro study to see how well do these vaccines that are based on the original strain of SARS-CoV-2 do against the newest BA.4/5. Unfortunately, we're not comparing apples to apples here because these are specific days after the vaccine or the infection have been given. And of course, they do the best job that they can, and they report that these are the days. Interesting to note that the Novavax, which is 82 days out, and the Johnson & Johnson, which is 79 days out, you can kind of see a difference in comparison there. Whereas the Moderna, which is 13 days, and Pfizer, which is 14, seem to have more of a robust response. It's interesting that Novavax is fairly similar to the Pfizer, even though it's about 14 days out. But it's really hard to make that comparison between vaccines. So finally, the last thing I want to point out, you've probably seen this already with this graph, is simply that each one of these vaccines represent the full dosing of the vaccine without boosters. And so the question here in 2022, summer going into fall and then finally into winter time, if you have been fully vaccinated and you're considering getting a booster, the question is why would you get a booster that is based on an old strain or variant of SARS-CoV-2 which doesn't exist anymore to try to protect you against a variant like BA.5? Is it going to be beneficial, even with just neutralizing antibodies? Let's take a look at the data. That's exactly what they decided to do in this study out of University of Washington. So let's take the scenario again of I've had 2 shots of either the Pfizer and or the Moderna, and the question is should I get a third booster of Moderna or Pfizer at this point? And so they actually looked at that. Here we have, again, the regular infection. Here we have Moderna at 2 doses and Pfizer at 2 doses. And then what we see here is about 30 days out after the 3rd dose of either Pfizer or a Moderna. So this could be a Pfizer, Moderna, or a Moderna, Pfizer. What are the neutralizing antibody results at this point? And you can see clearly here that whereas this was 2, that this is 3. Let's look at the BA.5 because that's the question that we have. And clearly you can see here that there is an improvement. Even though this is a 3rd shot of a vaccine that's based on an ancestor of BA.4 and 5, there's still a benefit specifically against the BA.4/5 subvariant. You can see that in all of those cases, that you go from fairly low neutralization titers to a fairly higher neutralization titer, even though you're not using a vaccine that is based on BA.4/5. Why do the neutralization antibody titers get lower with time? That might be a disadvantage, but in fact, it's an advantage. You want this to happen. Imagine an immune system with every antigen that it ever saw in its entire life would continue to pump out in the same amounts and the same quantity, the same amount of antibodies that it did. Eventually, your body would be filled with antibodies. It has to be able to decide which antibodies are important to produce and which antibodies are not important to produce. So long as they have the memory cells ready to produce at will, you're going to be okay. That's the whole point of a vaccine, is to allow your immune system, which is so complex and such an amazing system, the ability to see ahead of time the viral proteins so that at a moment's notice, if there is a re-challenge of that viral protein, the immune system can then wake up the cells that are able to make the antibodies and to make them quickly. When the immune system doesn't have that foreknowledge, it takes a long time and the virus gets a head start. That's the whole purpose of a vaccine or being infected previously. Let's go to another example here. How about if you've had 2 shots of Novavax? In the United States, we haven't even had the opportunity to get vaccinated with Novavax because it's not available. But there are people outside of the United States where Novavax has actually been approved. And the question is if you got a 3rd dose of another type of vaccine, either Moderna, Pfizer, or J&J, what would that do specifically to your neutralization titers against BA.4/5, even though you're getting a vaccine which is based on an ancestor of that subvariant? And the answer is, as you can see here very clearly, that neutralization titers go up with a 3rd dose. What about Johnson & Johnson? You've had 1 shot of the Johnson & Johnson, and the question is should you get a booster, either with J&J or Pfizer in this case, is what they looked at. And in both of those cases, significant improvement in neutralization titers in patients who have had 1 shot of J&J. And you can see here the mean days after the event that we're looking at. In this case, it was 14. What about AstraZeneca? Here we have 2 doses about 30 days after, and we vaccinate with either Moderna or a Pfizer vaccine. And what happens? 87 days later, instead of having neutralization antibodies in this range, we have it in this range. I think this is actually really good information because things are so diverse now with people getting different types of vaccines. And the question is, what should I do? Is it going to be an improvement, especially as we're now going into the fall and when the daylight hours are becoming less and less? And we've talked a lot about things that you can do that have nothing to do with the vaccines that can improve your innate immunity. And we've talked about getting out into the sun. We've talked about making sure you're getting enough sleep. We've talked about making sure that you are exposing your eyes to bright light in the morning to keep your circadian rhythm in check and making sure that you're not exposing your eyes to bright light after 9 o'clock at night. We've also talked about hydrotherapy. We're not afraid to talk about these things that can actually improve your overall health, but I think a lot of people are interested in knowing about what's going on with the different types of vaccines and also this variant that's going around. So what about Sputnik? And there's not a lot of people here in the United States that have had Sputnik. I'm sure there's some people around the world that are watching that may have a question about that. Here we see 60 to 90 days after 2 doses of Sputnik, we're seeing a reduction in the neutralization antibodies. What happens if you add to those 2 shots of Sputnik either an AstraZeneca or a Pfizer vaccine? You'll see there that there is an increase in the neutralization antibody titer. What about Sinopharm? This is also a vaccine that is very popular in Asia. 2 doses of Sinopharm, 69 days later, on average, what do you see here? You're seeing antibody titers in this range. If you were to add a Moderna or a Pfizer to that, you would see that there's a significant increase there in terms of neutralization of the antibody titer. So I found this interesting graph that they put in the paper, and this is looking at neutralization versus days post-symptom onset or the last dose of their vaccine course. You can see here the different vaccines and infection here. Infection is the green dot. You can generally see that as time goes on, regardless of what you're talking about, those antibody titers are going to get less and less. And again, here specifically, we're not actually measuring antibody titer, but we're measuring the amount and the quality of these antibodies to neutralize the spike protein. Just want to make that clear. But we know that in the human body, that antibody titers do get less and less over time. And so it's not surprising to see this. Again, we said the reason why that would be the case. Again, we can't keep antibodies at the same level for the whole time for every single antigen that the body ever sees, because then we would have just too many antibodies floating around in our circulation. One of the things I think was interesting, that it made me go back and reflect a little bit, is looking at the natural infection. I remember Dr. Crotty in an interview on MedCram talking about some of the issues regarding vaccination versus natural immunity, as it's often called. So we're looking at just a regular infection. And as you can see here, the one thing that sort of distinguishes this previous infection from the antibodies that are created from these vaccines is that we see a lot of very low neutralization levels here with previous infection. And we really don't see that with any of the other types of options here in terms of the vaccines. And if you think about why that might be the case, you can imagine that there's some people that could get SARS-CoV-2 positive, but they might have a very, very mild infection because they got a very small inoculate. In other words, enough to make them positive, but not enough to really create a good immune response. You can imagine that the immune system, if it gets just a very small challenge, might not make that robust enough response to give you immunity to the next infection. You can imagine that there is what we call heterogeneity there with that. Whereas with the very standard doses of a vaccine, you're going to get a high enough dose that in most cases your body is going to have a response to that.
The nuance to that is that actually people who have been infected with COVID-19, their immune memory is very heterogeneous. There are a lot of differences from person to person. And so we noted that, other studies have noted that, where if you look at the data in our paper, whether you look at the antibody measured or the B cells or the different kinds of T cells, there's about 100-fold range between any 2 people at sort of opposite ends of the scale. So that's a big difference, right? So we can say, yes, overall, the vast majority of people have measurable memory, and it looks like decent memory. But if you look at specific individuals, there are big differences and 100-fold differences. And as I said, we don't know what exactly is needed to protect against this virus. But certainly 100-fold difference is big, right? If you were playing a basketball game, right, and one team scored 1 point and the other team scored 100 points, that's not very close. And you would say there is a difference there that you should pay attention to. And immunology is not a basketball game, but still, right, that's a large difference, and we don't know how to interpret those details. And so, in fact, we said, probably it's plausible that people at the low end of that are more susceptible to reinfections because it looks like they might have a lot less immunity than some other people. And unfortunately, there's no simple way to predict which person is which, right? So we can say at a population level, yeah, most people have measurable immune memory. But some people, something like 5% of people, are really at the low end. So are those people at greatest risk? And so that's where the immunology is. If you put on your hat as an individual, or if you put on your hat as a public health professional, right, you're gonna view those data in some different ways, right? So at an individual level, one end of the spectrum would be I myself am comfortable getting infected. I don't mind if I get infected. I don't mind if I get sick. But I don't want to end up in the hospital. That would be, maybe one end of the spectrum of a person's level of comfort. How protected can I expect that I am having already had COVID-19? It's quite rare for somebody who has previously had COVID-19 to end up hospitalized. The numbers are so low, we can't even get good numbers still at this point, I think is where that is. But that you'd probably want to warn the person, that look, even a non-hospitalized infection can be pretty serious, right? Long COVID issues have been present in people who don't get hospitalized. And I'd say another end of the spectrum would be somebody saying, no, I don't want to get sick at all, either because of the age category they're in, right, or underlying conditions, or just that they don't want to get infected and be passing it on to other people, right? Whether or not they themselves would be healthy, they don't want to risk their parent or grandparent or neighbor, right, getting infected by them. So they really don't want to change their behavior unless they have a very high level of protection.
Now what we're looking at is the very same graph that we saw before, except now with the booster. So instead of 2 shots of Moderna, we're looking at 3 shots specifically. Instead of 2 shots of Pfizer, we're looking at 3. And again, all of the other types of options that we talked about. And you can generally see the types of neutralization that we're looking at here, and specifically how things go post-symptom onset or the last dose. We're actually seeing it holding up a little bit more and less of a reduction across the board. And this led the authors of the study to say administration of a homologous or heterologous booster based on the Wuhan Hu1 spike sequence markedly increased neutralizing antibody titers and breadth against BA.1, BA.2, BA.2.1, and BA.4/5 across all vaccines evaluated. Our data suggests that although Omicron sublineages evade polyclonal neutralizing antibody responses elicited by primary vaccine series, vaccine boosters may provide sufficient protection against Omicron-induced severe disease. And for those of you who are interested, I list here the funding, but that's also available at the research article, which we'll post and give you the link in the description below. Now, I've heard a lot of people ask the question regarding all of this spike protein stuff is why do we formulate vaccines specifically against the spike protein, something that is mutating so often? Why don't we go against some other surface protein? Even a year ago, when we had Dr. Shane Crotty on, he answered that question really well. So I will go ahead and refer to what Shane Crotty says here.
Why did both companies choose to use spike protein for their target for this vaccine?
Right. So there are about 29 licensed human vaccines, depending on how you count, and almost all of them work on the basis of protective antibody responses. And so when you're trying to move fast with vaccine development, the most obvious target is to try and make antibodies against the protein that's on the surface of the virus, because antibodies work by binding to the surface of a virus and essentially covering the virus and keeping the virus from doing anything. That's really the simple way to think about antibodies working. And so for previous coronaviruses, it was known that there are a couple of different proteins on the surface of the virus. But it's really the spike protein that's the major one and probably the most important antibody target. And sure enough, in the months subsequent to those decisions, lots of data have accumulated that have said essentially all of the neutralizing antibodies, the important antibodies against SARS-CoV-2, are against the spike protein. So the spike protein is the best target to focus on for antibodies. And when I talk to you about, there really being three parts of the immune system, one of the concerns has been that, well, sometimes antibodies aren't that great at stopping all viruses, and then you really need the T cells to kick in. And the T cells don't necessarily recognize spike. They might recognize some of those other 25 proteins. And so that was actually our first major scientific study on COVID-19, was to ask in infected people, do people make T cells that recognize spike also, or only other proteins? And what we found was that in infected people, actually people make a lot of T cell responses to spike also. And so that was a good sign supporting the vaccine development at all, that if you are some viruses, you have to choose more than one protein. For this virus, it's looked like, just choosing one protein is a reasonable way to try and get antibody responses and T cell responses.
So again, I thought this was a very interesting article, as it answers a question that a lot of patients have been asking me. They understand that there are specific vaccines in development to attack the specific spike protein from the BA.5 or Omicron variant. And that actually may be a good idea. It may improve what's going on. But the question is, should you wait for that to come out, or should you take advantage of the boosters that are available now if you're interested in getting that type of protection? And in conjunction with the fact that we here at MedCram followed the Swiss cheese model, which essentially says is that we're looking for anything that may be beneficial to protect people from getting disease and ending up in the hospital, whether that be improving their own innate immune system through natural means or taking advantage of the available vaccines to protect them. Then we're going to review the data and make that available for everyone so they can make their own decision. And with that, I'd like to thank you for watching this video. Please subscribe and turn on notifications. And for those that are interested in getting more information and continuing medical education for healthcare providers, join us at medcram.com. Thanks for joining us.